Can you see?
Yeah.
Okay, so how do today we will be talking about some resources we're going to be using, not just for research, but in general for like researching about genomes. And so today we'll be learning about the UCSC Genome Browser. So if you could go to this website. Oops. How do I? Oh, this website. Okay. So this is the University of California, Santa Cruz Genome Browser. And if you go here, you can see it'll give you like, you can choose a species and then it'll give you like the gene sequence for it. Vidi, is that right?
Yeah.
So here I put in SARS-CoV as an example, and then we can select, this is like the placement of it. So if we select COVID, then we'll be able to see the, like the, what's it called?
You can see like different genomes, obviously, and then also like the spike proteins. You can see different mutations, proteins, receptors, like everything that's on whatever you're looking at. So in this case, like COVID, on the bottom, you can see like the different mutations. So
yeah,
you can see like Omicron,
we can see
Alpha, Delta, and then you can see like specifically what proteins are
yeah.
in them.
So in COVID, the S protein, which is a spike protein, is mostly for, I think it's mostly for what um makes it contractible. So you can see at the S part here, all the like mutations here are mostly in the S protein. So that's why the COVID was more mutable, and that's why there were a lot of mutations in the S protein. And then, am I missing anything, Vidi?
No, I think that's good.
If you like go into the specific proteins, you can see it gives you like the name, and then you can also see like the proteins. Is it that the amino acids that make it up?
Yeah.
I think so. And then the sequence. This is like the whole sequence for that too.
For the
So
S protein. Yeah.
yeah. And of course, you can also find more on like the S glycoprotein. There's more on that too, and it has more data on like the function of it.
Basically, we can use this browser to help us look at, like, for example, if we're looking at a specific mutation in COVID, we can take a look at, I guess, how the spike proteins have mutated, what proteins are a part of it, and things like that. So it's like more information about whatever you're trying to look at.
Mm-hmm. Okay, so in the slides, I basically have, the information for this and what you're supposed to type in and just seeing the mutations and stuff. And then here we also have another website that's good for visualizing like genomes. So with COVID as an example, you can go to the sequence resources and then you can see view record. And this will basically give you a similar graphic, but in this one you can also see the specific nucleotides and, why is it not loading? Okay, and these proteins with it. So if you see here, if you zoom in, I believe, let's see. No, that's not it. Vidi, do you remember how to show the nucleotides?
Okay, give me a second.
But in here, let me read. You could also find the S protein like this. And then it will show you that right here. And then, oh, it'll show you like the location of it in the gene position, and a lot of other stuff. Okay. Oh, is it this? No. Wait, Vidi, do you remember the drop down thing? I remember there was like a drop down thing where they showed
I
you
know what you're talking about.
for the nucleotides.
Yeah, I forgot where it was too. I can look back if you want.
Oh, I think it's this. Look at that.
Yeah, you got it.
Look how cool. Okay, so like these numbers, oh no, these letters, oh my goodness. These letters represent like the nucleotides that are in the S protein. And then here you can also see like the TAT, you see that. And then down here, there's also TAT. And below that Y, that would be the amino acid that stands for. And the letters down here should also match with the letters over here because they're from the same protein. Okay. And then another resource we can use for COVID visualization is NextStrain. We didn't use this that much in MIRCOR, but it was interesting just to see like the graphics of it. It's not loading. Okay.
It'll go.
Right. My computer's a little slow. Okay. But if you scroll down, then you'll see just the graph of all the variants in the world. And down here, you can see like the y-axis is the dates, and then the percentages, and you could see like the percentage of each variant that's spreading. Like right now, Omicron is more widespread than Delta. Well, like in October, mostly it was Delta that was spreading. Yeah. Okay. This was our presentation on the research on COVID, which will be, I think will be in the Genes and Health contest. Right, Vidi?
Yeah, so this is useful for the Genes and Health contest. It's also just generally a good tool to know how to use, especially when you're looking at different proteins and genomes. And yeah, this one was like a shorter presentation, but does anybody have any questions? Okay, then was this the end, Grace?
Yeah, we can also go more into the research part, but do we want to do that today?
How long do you think it would take?
Oh, I could just share. I was working with my miracle group and I could just share.
Oh yeah, sure. We could do that really quickly.
Okay. Hopefully my group won't mind. Okay, I'm gonna stop sharing first. Where'd it go? Oh, I don't know how to use my computer. Oh my goodness. Okay, so last week, wait, Vidi, do you want to summarize last week while I pull it up?
Yeah, sure. So last week we went over KEGG pathways and string DB. Um, so basically what we did is we took all of the genes that we had in string DB and we looked at specifically genes that were part of like the proteasome like pathway. And so we took those genes and we put it into KEGG, and then we looked at how they're in like the neurodegenerative disorder pathway in KEGG and how they're like related to one another. And yeah, and
Yeah.
then yeah, I think we went over string DB twice, so I think it should be.
Yes. Yes. So let me share. Wait. So my group basically in MIRCOR, we went over the specific like cellular processes and KEGG pathways and identified like which genes were significant in it and if they were downregulated and upregulated and what like it would mean for the patient. So let me see. Why can't I find it? Okay, give me a second. Okay. Okay, here it is. Let me share. So last week we did the string DB for the specific genes. String DB. I can do this again. So if you didn't know, if you forgot, oh no. Okay, we copied this. We copied this row. Oh my goodness, my computer is so slow. It's not loading. Okay. It's loading. Okay. And then this was our string DB map. So after we analyzed it, we had like biological processes, and I was focusing on the cellular amide metabolic process. So that was all the red dots, and we just copied all the genes that were significant here. And then I did a specific research on one of the genes, which was PDHX, and that was a pyruvate dehydrogenase protein X compound, and it was required to bind to other, I think, proteins in the pyruvate dehydrogenase complex of eukaryotes, and the binding is important for the functional PDH complex. So I did some research on this, and according to the table, if like I search up PDHX, that was this column, and the log FC for that, this was a log FC. So that was negative. So that means it was downregulated compared to the control group. So downregulation of the PDHX means that it inhibits the function of the PDH, which would probably be a cause of bipolar disorder. And I found an article explaining like connecting the PDH complex to bipolar disorder, which could be another area of research that you could explore. But yeah. And then our group also did further research on like cellular components and the KEGG pathways, like the prions disease, since most of these genes that were affecting bipolar also were downregulated compared to like those in the prion disease. Okay, so that means that these genes had a correlation, and maybe that these two diseases also have a correlation. And that's the same with like Huntington's disease, Parkinson's disease, like all of these genes were all downregulated. So that's why these have a connection to the genes here, like as seen here. And yeah. Viri, I think we're good.
Yeah, that was also a good review of last week's stuff. So does anybody have any questions from this week, last week? Anything about like StringDB or KEGG or the Genome Browser? Okay, if not, then and then also if any other questions come up, you can just message us, or you can message in like the general channel in Discord, and we can help you answer any questions.
We do attendance.
Oh yeah, let me send the attendance.
Okay, I got the where's the link?
Here, I got it.
Okay.
Okay, here is the attendance form for this week. What do you want to make the code word, Grace?
What? I don't know. You can make it.
Okay. The code word is just put Genome Browser.
Okay.
Okay, and then when you guys are done filling it out.
Wait, one word, Genome Browser?
Yeah, it doesn't really matter.
Okay.
Yeah.
Thank you.
Yeah, thank you.
Thank you.
Thank you.
Okay. Viri?
Yeah.
Hello.
Hi.
Is it binder going okay?
Yeah, let me pause the recording.